Hello, my name is Carson, and I'm doing the Kras protein in the G12C mutation for my project. To begin, Kras is a small GTP-based protein involved in cell signaling that regulates cell growth, proliferation, and survival. And Kras belongs to the Ras family of proteins, which functions as molecular switches controlling key cellular pathways like MAPK. Moving on to cell location, Kras is anchored to the inner side of the plasma membrane via a prenyl group attached to a C-terminus and interacts with upstream receptors and downstream effectors to transmit growth and survival signals. So, cell type, Kras is widely expressed in many cell types, including lung cells, colon cells, pancreatic cells, and so on. And its connection to cancer, the disease listed, the Kras G12C mutation is found in approximately 3% of colorectal cancers and 1% of pancreatic cancers. The G12C mutation results in consecutive activation of Kras, which causes uncontrolled cell growth and tumor formation, leading to diseases such as cancer. So the normal functions of Kras has cycles between two states, the active site when bound to GTP and the inactive state when bound to GDP. Mechanisms. In the inactive state, Kras binds GDP. A genuine exchange factor stimulates the release of GDP and the binding of GTP. When GTP is bound, Kras undergoes a conformational change, enabling it to activate downstream signaling proteins. A GTPase activating protein stimulates hydrolysis of GTP back to GTP, returning Kras to its inactive state. This results in tight regulation, which allows self-growth and division. Now, moving on to the mutation, I'm going to cover up glycosine, as seen there, with the new mutation after. This is the mutation with cytosine. Hope I'm pronouncing that right. And this represents the stress in the locked-in state that the cell endures. So, mutation at codon 12 obviously replaces glycine with cysteine. Effect on the protein function. The larger cysteine residue introduces steric hindrance. The tithyl group forms adherent disfluid bonds and local misfolding of the P-loop. And gap-mediated GTP hydrolysis is impaired. And Kras becomes locked in the active state, which is constant downstream signaling. And there's going to be increased activation which leads to uncontrolled self-growth, resistance to apoptosis, and cancer progression as well. So, moving on to the structure yet again, the red part of my model is the primary 1 structure, which is a linear sequence of 109 amino acids. Glycosine at position 12 is critical for the flexibility of the P-loop. Moving on to the secondary structure, which is the helixes, which is represented by the green pipe cleaners. There's the alpha helixes, which make up about 40% of the Kras structure. And there's also the beta sheets, which is the white part right here, which is the formed core of Kras. And there's also loops as well around, drawn on, which connect helices and sheets. And the tertiary 3 structure is the global structure, which is also the red as well. Is formed by the folding of helices, sheets, and loops, and stabilized by hydrogen bonds and hydrophobic interactions. And as far as the quaternary structure, Kras functions as a monomer, so it does not have a 4 structure. So impacts in the mutation of the structure, it's a steric clash, formation of adherent disfluid bonds, and reduces interaction. And how it affects the organisms is tumor formation and so on. And 3 limitations is size difference, dynamic flexibility, and complex interactions. So summary, Kras is a key regulator of cell growth and survival. The G12C mutation replaces glycosine with cysteine, introducing structural rigidity and preventing GTP hydrolysis. This locks Kras in a consecutively active state, driving uncontrolled cell growth and cancer progression. Thank you so much, and I hope that you enjoyed the video.